So most ATP is generated in the mitochondria. This requires oxygen as the final electron acceptor. In the presence of oxygen, we'll see that pyruvic acid enters the mitochondria and is oxidized by aerobic respiration, where we'll capture some more of the energy of the original glucose. There are two principal steps for aerobic respiration: the matrix reactions and the membrane reactions. The matrix reactions are where the enzymes are in the fluid of the mitochondrial matrix. These will be the Citric Acid Cycle, Krebs cycle, or TCA cycle, all the same thing. Or the membrane reactions, where the controlling enzymes are bound in the membranes of the mitochondrial cristae, so the inner mitochondrial membrane. This is where we'll see the electron transport chain and oxidative phosphorylation. So before we actually get into the citric acid cycle, there's a little bit of modification that we need to do to pyruvate. So pyruvate, or pyruvic acid, needs to be converted into acetyl-CoA. So first of all, we'll see the decarboxylation, so removing one carbon. So now we're down from 3 carbons to 2 carbons that will be acetyl-CoA. Then we have reduction. Reduction is where one of the NAD cars is going to come along and pick up 2 electrons and 2 hydrogens, which are high energy, and they're going to be transported off to the PROM, or the electron transport chain. Then the acetyl group will bind to coenzyme A. This is a derivative of vitamin B. So acetyl-CoA is then in perfect position to enter the mitochondrial matrix and the citric acid cycle. So remember, how many carbons did we start out with? It was a glucose, and a glucose has 6 carbons. So how many pyruvic acids came from the breakdown of glucose? Yes, 2. If 2 pyruvic acids come from the breakdown of glucose, then this whole system will have to run 2 times, right? So we multiply everything in here by 2. So we can have 2 times the pyruvic acid, which leaves us with 2 times acetyl-CoA, and then 2 times everything else that happens in this cycle. So at this point, now that we've deep carboxylated and reduced our pyruvic acid into acetyl-CoA, how many of the original carbons of glucose are left? 2 from each pyruvic acid, and there were 2 pyruvic acids per glucose, so we have 4 of the original carbons of glucose left. We've also released some electrons and hydrogens, high energy compounds, ready to be traveled off to the electron transport chain. How did we lose the carbons? Where did the carbons go? Yeah, you're right. They're going to be the carbon dioxides that are exhaled in each breath. So now let's take a closer look into the citric acid cycle. As we know already, acetyl-CoA enters the citric acid cycle. Now, this cycle is pretty much like a workbench. The key here is oxaloacetate is the workbench. And to oxaloacetate, we're going to add our 2 carbons of acetyl-CoA. And that reveals a 6-carbon compound called citric acid. As we go through this cycle, we're going to chop off some more carbons. Here we'll release a CO2. And again here we'll release another CO2. Those are the 2 CO2s that are left for each of the pyruvates. Again, this cycle goes around 2 times per glucose molecule. So the 2 carbons of acetyl-CoA get snapped onto the workbench of oxaloacetate. They become citric acid, hence the name of the cycle. Now we're going to see that water becomes removed, and we see that NAD comes in to reduce the sugar further and take electrons and hydrogens. We lose a carbon dioxide. Now we have a 5-carbon substrate. 4 carbons of it were the workbench, and 1 carbon of it is left from acetyl-CoA. And then the cycle basically repeats. We remove another free carbon dioxide, and we remove some more electrons and hydrogens in NAD cars, which will be carried off to the electron transport chain. So at this point, we're back to our original 4-carbon workbench in a slightly different form than oxaloacetate. But the next steps are just going to recycle the workbench back. So we've got 4-carbon oxaloacetate to attach acetyl-CoA to, and so on and so forth. Release carbon dioxide, release another carbon dioxide. Poof, there go all the original carbons of our glucose. As this 4-carbon chain is recycled, we'll release some energy from it, eventually in the form of ATP. But it goes through an intermediate step where GDP, guanine diphosphate, which is very similar in structure to ATP, becomes phosphorylated and becomes GTP, and then GTP passes its phosphate onto ADP, allowing it to become ATP. So we've made 1 molecule of ATP for each pyruvic acid that came into the system. We're going to see that we remove 2 more hydrogens and 2 more electrons with FAD cars, so they become FADH2. We're going to add some water into the situation, and then NAD is going to remove the final 2 electrons and hydrogens so that they can carry these high energy molecules off to the electron transport chain. So now we've regenerated oxaloacetate, so acetyl-CoA can join it and the cycle can repeat itself. Again, we had 2 pyruvic acids come in for each glucose we start with. That released 2 acetyl-CoAs to go into Krebs cycle. And thus Krebs cycle essentially has to run twice in order to release all of the original carbons from our glucose molecule. So again, the most important thing here is to track 3 things: carbons, electron carriers, and ATP. So how much are made in each step of this process? What happened back in glycolysis? Did we lose any carbons? Did we gain any ATP? And how many electron carriers were there? So let's skip back there for a moment. Here, there were 4 ATP produced, but remember 2 were consumed in the beginning to get the whole thing going. So we have a net of 2 ATP from glycolysis. And then we had some energy from the original glucose is contained in ATP, and some in NADH as electrons and hydrogens that are very excited. So we have 2 NADHs that were produced, and those we'll see later. They'll release their energy elsewhere to create ATP. We had a net of 2 ATP, and we had 2 pyruvic acid. So we didn't lose any carbons in the process of glycolysis because these 2 pyruvic acid are 2 3-carbon compounds. So now let's look at what additional products we gained in the process of the citric acid cycle. So here in the citric acid cycle, so we actually have a total of 6 carbon dioxides. All of the original carbons have now been oxidized and released as CO2. Remember, 2 of them in the conversion of pyruvic acid to acetyl-CoA, and then the other 4 in the citric acid cycle itself. We also have released 2 ATP per glucose in the citric acid cycle. and 8 NADHs and 2 FADH2s. So these are the products of pyruvate oxidation and the citric acid cycle. Note that all the carbon atoms of glucose are now gone and they're exhaled as CO2. We've lost some energy as heat, we've stored some in ATP that's ready to go, and then we have 8 reduced NADHs and 2 FADHs. Now these molecules are carrying the high energy electrons and hydrogens to the electron transport chain. So right now they're the kids in the cars ready to go to prom. And then they'll come and be released and converted into ATP at the membrane reactions. Now the citric acid cycle not only oxidizes glucose metabolites, but it's also a pathway and source of intermediates for the synthesis of fats and our non-essential amino acids. We'll illuminate the connection between the citric acid cycle and the metabolism of other nutrients later on in the chapter. So take a moment now to diagram the oxidation of pyruvate and the citric acid cycle. Consider just the main participants: oxaloacetate, citric acid. So adding the 2 carbons of acetyl-CoA to the oxaloacetate workbench. Note where the carbons are removed, where electrons and hydrogens are removed, where ATP is removed. So following the matrix reactions, we've got a lot of NADH and FADH2 carrying electrons towards the electron transport chain. We've released a little bit of ATP and we've oxidized all the carbons. So the purpose of the membrane reactions is twofold. First of all, to oxidize the NADH and FADH2 and transfer their electrons into the electron transport chain so that we can release their energy to produce ATP. The other purpose is to regenerate NAD, empty out the cars, so that they're available again to go pick up electrons at earlier reaction steps. So the mitochondrial electron transport chain is a series of compounds that carries out a series of membrane reactions. Most of them are bound to the inner mitochondrial membrane, and they're arranged in a precise order, which allows each protein to receive a pair of electrons from the member to its left. It passes the electrons down the chain to the member on the other side. As it passes it down the chain, the electrons will lose energy, much like if you take a hot potato and pass it from one person to the next, to the next, to the next. It will have less energy in the form of heat in it by the time it reaches the end of the chain. So there are many members of the electron transport chain. You don't need to memorize these per se, but they're grouped in enzyme complex groups. So these two here are in complex 1. They're at a higher energy state. They take the electrons from NADH and they pass them down the chain to enzyme complex 2. Coenzyme Q accepts the electrons from FADH at a slightly lower level than the enzyme complex 1 accepts electrons from NADH, and we'll see the implications of that later. Either way, the electrons bounce down this chain from one carrier to another and are finally received by oxygen, or 1/2 of an O2, at the end of the electron transport chain, where they will come together with hydrogen in order to form water. So as the electron transport chain receives electrons from our electron carriers, NADH and FADH2, the hydrogen atoms are split and they are pumped into the intermembrane space, so the space between the inner mitochondrial membrane and the outer mitochondrial membrane. We'll take a look at this shortly. The electrons travel down the chain in pairs, noted as 2e-. Each electron carrier becomes reduced when it receives the electron pair and oxidized again as it passes that electron pair along to the next carrier. Again, oxygen is the final electron acceptor. Each oxygen atom is going to accept 2 electrons from the final protein in the electron transport chain and 2 protons from the mitochondrial matrix, which will form water. This is the body's primary source of metabolic water. That's the water that's synthesized in the body rather than consumes. And it explains why our body absolutely requires oxygen. If there's no oxygen, the cell can produce too little ATP to even sustain life. So now the question is, how is the ATP actually produced? And it comes through this chemiosmotic mechanism. These are enzymes, again, that are located in the inner mitochondrial membrane. So if we look at the mitochondria here, the red membrane is the outer membrane, and then there's the inner mitochondrial membrane. There's a tight space between the inner and outer membrane, and this space will allow for a very strong hydrogen ion electrochemical gradient, as you'll see. Now I have an analogy we came to before. It was about the prom. NADH and FADH, cars full of excited electrons and hydrogens. The hydrogens are the boys and the electrons are the girls. Remember that one? We're going to take it a few steps further. So now we're at the prom. So here we're in the mitochondrial matrix, and embedded in the membrane is this electron transport chain. The green, yellow, and blue enzymes here, complex 1, 2, and 3. And as we arrive at the prom, the NADH plus H plus cars are coming in to drop off their kids at the prom. They drop off girls and boys. It turns out at the top of the stairs, there's a bouncer. And the bouncer, he is really kind of a nasty guy. So he takes the girls, or the electrons, and says, oh yes, you come on down the stairs, and they go down the stairs and down the stairs and down the stairs, and they end up going into the prom, or into the prom over here. However, the guys come to the bouncer, and the bouncer throws them out into the back alley. So there's this tight space in the back alley. It's the intermembrane space. So as electrons are transported down the chain, hydrogens get thrown into the back alley here in the intermembrane space. So there becomes a very steep gradient of hydrogens, and they're pissed off. So there's a lot of energy there. They really, really, really want to get back to their girls. So they hang out back here getting mad for a while, and then they find the back door, and the back door is like one of those revolving doors. And the hydrogens will rush back in through the back door, spinning the door so fast and releasing so much energy. And they're finally back into the prom. They go over and they meet up with their electrons that have bound with water. Water's like the caretaker. Once the girls get inside the prom, he's like, oh yes, ladies, I'll take care of you until your guys show up. And so the guys show back up, and they're all reunited. Everyone's happily ever after. We've got the formation of water. And as these hydrogens whiz through this spinning door, we'll see that the energy is captured and phosphorylates ADPs in order to make ATP. So the chemiosmotic mechanism following the electron transport chain is the prime site for ATP synthesis in the process of cellular respiration. So let's look at it again. This back door, the spinning door, is called ATP synthase because it synthesizes ATP. This hydrogen ion current rushing back through the ATP synthase channels is what drives the ATP synthesis. So NADH and FADH2 will drop off their electrons at the electron chain, the staircase, and they bounce down from one to the other, and finally they're received by oxygen, which is a 1/2 O2. So that's just an O all on its own. It's split oxygen. And then the hydrogens that are whizzing through the door come along and meet with the electrons that have found the oxygen, and there's where we have the production of water. So this is it. This is the key place where water is produced and oxygen is consumed in the process of cellular respiration. I know the analogy is kind of goofy, but it works for me. So take a moment now and see if you can diagram this process. Now I like to draw just a very simple staircase in the membrane and understand where the electrons are drawn in and they bounce down the chain and they finally meet up with oxygen and their hydrogens to form water. See what you come up with. We'll take a look at it in class. So NADH releases an electron pair to the electron transport system, and it bounces down the chain, and by the time we have hydrogens coming in through the ATP synthase, we're going to produce 3 ATP. Now FADH releases its electrons to proteins further down the electron transport system, and so they only release about 2 ATP. Complete oxidation of glucose to CO2 and water produces 36 to 38 ATP. So when we consider the ratio of energy output to input, we've got an efficiency rating of about 40%, where the other 60% is lost as heat. So it's not a very efficient system, but it's much more efficient than running just glycolysis. Here's a really good figure for you to summarize where all the different pieces are coming from. This is the one that I'd like for you to produce for our diagramming assignment this week. We've got glucose going through glycolysis. What does it produce? Pyruvate is oxidized, and then we have the citric acid cycle. What are the products of this as far as carbons, ATPs, and electron carriers? Over here we can see the ATP totals for the whole process of oxidative respiration. Finally, we take a look at the glycogen metabolism. So glycogen is a storage carbohydrate. ATP is used very quickly after it's formed, so it's considered an energy transfer molecule, not an energy storage molecule. So if there's plenty of ATP, then we need to store the glucose in other compounds, such as glycogen and fat. So glycogenesis is the synthesis of glycogen. It's stimulated by insulin, and it brings glucose monomers together in a more complex form so that they can be stored. Glycogenolysis is the hydrolysis of glycogen. Glycogen releases glucose between meals, and that's stimulated by glucagon and epinephrine. Only the liver cells can actually release glucose back into the blood. Remember that molecule, glucose 6-phosphate? The phosphate prevented it from leaving the cell? Well, the liver cells, remember, have a special enzyme called glucose 6-phosphatase, which can remove that phosphate group and release the sugar out into circulation. Gluconeogenesis is the synthesis of glucose from non-carbohydrates, such as glycerol and amino acids. And again, this occurs mostly in the liver and sometimes in the kidneys if absolutely necessary. The figure below here sort of summarizes that. Glucose 6-phosphate, we can convert that into blood glucose with the action of glucose 6-phosphatase. Hexokinase will bring blood glucose, as you know, from the beginning of glycolysis back to glucose 6-phosphate, and then we can go on and produce glycogen if necessary. So that brings us to the end of this section on carbohydrate metabolism. Now you're ready to move on to lipid and protein metabolism.